Sir, good morning. Good morning, sir. Good morning, sir. Sir, am I audible? Good morning, sir.
Good morning, madam.
Good morning, sir. Am I audible, sir?
Yes, madam, you are audible.
Okay, sir.
Yeah. So maybe Kanchan Koili madam will be joining later by 10:30, 10:45. She will join.
Okay, sir.
Yeah. By the time your topic comes to little bit finishing mode, she will join.
Yes, sir. No problem.
Yeah. So we will be starting in another maximum 7-8 minutes, madam.
She is doing a
అంతవరకు మీరు అన్మ్యూట్ పెట్టండి, ఈకో వచ్చేస్తుంది. అన్మ్యూట్లో పెట్టండి.
Shall we start, madam?
Yes, sir. Yes, sir.
Sir, between the two speakers means now only two speakers are ready.
A very good
Yes, sir.
morning.
Yes, sir. Okay.
Yeah.
A very good morning to all. This is Dr. Monica Nijhawan from Gokaraju Rangaraju College of Pharmacy. I welcome all delegates and resource person to join the conference on recent approaches in herbal drug and cosmeceuticals on behalf of Gokaraju Rangaraju College of Pharmacy. Now, I request our Honorable Principal Sir, Dr. G.T. Kulkarni, to address the gathering.
A very good morning to one and all the delegates who have joined in Zoom, as well as those who are watching this program live streaming on YouTube. I would like to welcome all of you, and also today's two speakers. Once we introduce, all of you will know why these two speakers have been selected for today's program. And one thing which I would like to tell now is the first speaker, Dr. Harsha Khadakwal. I am very happy to say that she is alumnus of our Usmania University. So I would like to welcome Dr. Harsha, as well as the second speaker. Actually, madam will be joining little late, she has told me. Professor Kanchan Koili, who is from Jamia Hamdard, the best pharmacy education institute in India.
Yes.
So here, little bit introduction about Gokaraju Rangaraju College of Pharmacy. I would like to present here because many participants are there from different states of this country. So for the benefit of them, I would like to give little bit introduction about GRCP. Gokaraju Rangaraju College of Pharmacy started in the year 2003 under the aegis of the society, Gokaraju Rangaraju Education Society. And this society was started by a person who himself is a pharmacist, Dr. Gokaraju Gangaraju. Because of that, he knows the value of pharmacy education and the avenues of pharmacy education. So our society has many members who are pharmacists, because of which the pharmacy education in our society is thriving. And ours is the college which got first accredited in the undivided state of Andhra Pradesh. In 2009 itself, we got the first NBA accreditation. Until now, 5 cycles of inspections have been completed by NBA. Very recently in September, we had the last inspection. And also this year, we are in the rank 93 in NIRF. And research is one of the most important aspect of academics, as we know. And GRCP is always involved in research. From 2003 till date, nearly 1,30,00,000 funding has been obtained by GRCP. And presently, we are doing 2 industry-based projects worth nearly 10 lakh rupees. And from beginning till date, the college has more than 1,000 research papers. So this shows the research motivation of our faculty and our students. Presently, we are also R&D center recognized by Usmania University. Many of our faculty are guides, recognized guides in Usmania University as well as in other universities also. So with this brief introduction of college, I would like to welcome the speakers, the delegates, our faculty, our students. And here, few people are visible in Zoom. Many people are there in YouTube, and nearly 150 students are watching this program in our auditorium. So I'd like to welcome all of you, especially those maybe with a short notice also. Within 10 to 15 days, we got an overwhelming response when we called for the abstracts of this e-conference. Nearly we have accepted nearly 80 abstracts, which will be under 5 streams. We are doing that e-poster presentation in the afternoon session, and in the morning session, we have 2 lectures. So with this, once again, I would like to welcome all the delegates, participants, presenters, speakers, our faculty members and students to this one-day e-conference, which is being organized for the first time by GRCP. And today, presently, we have our past principal, who is continuing as professor and research coordinator in our institute, Professor CVS Subramaniam, who doesn't need an introduction. All the people in India have read his books only. So I would like to welcome CVS Subramaniam sir, and I would like to hand over the stage to CVS sir for introduction of the guest, and I would like to welcome him as chair of the session. Thank you, sir.
Thank you very much, Dr. Giriraj Kulkarni. It gives me immense pleasure to chair this particular session on these recent approaches in herbal drugs and cosmeceuticals. At the outset, on behalf of the college, staff, students, and the management, I welcome Dr. Harsha Karkwal to this particular webinars. Madam, we invite you to our college.
Yes, sir.
I happen to be a colleague of Dr. Pushpangadan, who has
Yes, sir.
the director in NBRA.
Yes, sir.
We were there together in RRL Jammu.
Okay,
We
sir.
shared the same office
Okay, sir.
and spent 4 years with him.
Yeah.
And when he was a director in NBRA, I visited that for inspections of scientists,
Okay, sir. Great.
selection committee.
Okay, sir.
Very close to here. Anyway, I
Pangadan sir is another gem of a person, sir. Down to
Yes,
earth.
sir.
Really.
Pangadan sir is a Padam Shri. He is
Yeah.
associated with Amity also for so many years, and I'm blessed to have his blessing since 2000, since 1999. So it's great to know, sir. Yeah.
before 2 terms in NBRA, he was there in TBGRA at
Yes, sir.
Trivandrum,
Trivandrum. Yes,
Tropical
sir.
Botanical Research Institute.
Yes, sir.
I also visited. In fact, I am happy to introduce
Yeah.
Dr. Harsha Karkwal, FRSC. She is presently director in MIT Institute of Phytochemistry and Phytopharmaceuticals, as a coordinator in MIT Center for Carbohydrate Research. She has more than 20 years of experience in research and analysis with leading research institutes, have research on various topics, including plant hydrocolloids, derivatization, bioactive molecules, pharmaceutical analysis of native as well as modified seed gums by using latest techniques. It's a cut-off-the-box thinker and demonstrated the abilities to meet the deadline and work under pressure. She is BSc from Usmania University, MSc from Commune University, and PhD from NBRA,
NBRA.
National Botanical Research Institute, Lucknow. She has a large number of achievements. I just read a very few for the paucity of time. Her total publications are 66 in national and international journals. She has research papers for the last 5 years, about 16. Total patents filed and published. Total filed is 75, published are 63. Patents granted is 17. She has total book chapters, 24 books. Total citations are very huge. Consultancy projects, etc. Completed several research projects. She has supervised PhD students and 2 PhD students are working under her for their PhD. In fact, she had a very long experience in MIT and started with a lecturer and rose up to the position of a director. That speaks volumes of her achievement in the field of pharmacy profession in general, and also MIT University in particular. These few remarks, without taking much of your time, I again invite Dr. Karsha Karkwal to this particular webinar, and I hand over the mic to Dr. Harsha. Madam, please speak.
Yes, sir. I think, sir, one of them is not muted. Josephine Joy, I think.
Yeah, Mr. Josephine, kindly mute yourself.
Sir, respected sir, thank you so much for such a wonderful introduction, and good morning to Kulkarni sir, to Dr. Subramanian sir, for inviting me for this prestigious conference. And me andarki na namaskaram. I love Hyderabad. I was born and brought up there, so I have a special I always feel that I belong to Hyderabad. So this is a anything for Hyderabad. It's like that. So today I'm going to cover a topic of bioactive components from herbal medicine, recent advances in treatment of osteoarthritis. So we all know that we are doing this is one of the health problem which is being they have taught both in allopathy as well as in traditional medicine, and I believe that if we can really go back to our traditional wealth which was always there, and look into that perspective, then especially for muscle pains, for any kind of bone inflammations and other deformities, Ayurveda and other traditional modes of treatment plays a very essential role. So we are also collaborating in Amity. If I give you a brief gist, we have around 13 universities across India, and sir was also there. Kulkarni sir was there in Amity before. We have 150 plus institutes and centers, 300 plus programs, and there are more than 30,000 students getting scholarships, either 50%, 100%, 75%, like that, and almost like 80 to 100% placement in almost all the domains. More than 1,500 patents have been filed by the faculty, because before publication, we believe that if we can protect what we are, whatever idea that we have, so we definitely patent that, and we have got more than 300 funded projects and more than 300 plus labs in various disciplines. So osteoarthritis, if we take this topic, according we all know that, it is a degenerative joint disease, and it is osteo means it is related to bone system. And since students are there, I prepared it like that also. And arthritis means it's a joint inflammation in which one or more joints are inflamed. So I just want to tell that recently, my doctor also underwent an autoimmune disorder. It was equivalent to arthritis only, and doctors were telling it's an autoimmune and it has been triggered. And we were giving her five painkillers, steroids, antibiotics. But at last, we went to cortical, and the branch is there, and it worked like the combination of herbs, the oils and everything. They reduced inflammation within 15 to 20 days, in which the allopathic doctors had told that it would take around 3 years. She has to be in complete bed rest. It was hip synovitis for her. The fluid had come out of the bone. So it was very painful for her, for us also. And I think though and we are working closely with All India Institute of Ayurveda along with Rosenberg Institute for osteoarthritis, in which we'll be working on Vitex, the Godot plant. And in that also, we are looking out alternatives or bringing out something novel in the field of osteoarthritis. So there is constant progression, but definitely we are looking back into the traditional wealth that we all possess. So it's a condition in which one or more joints are inflamed, and it involves the loss of cartilage. And it is we know that it is characterized by bone lesions, by cartilage degradation, and there is inflammation. And around the world, it affects around 6% of population, and its frequency increases with aging. And especially after COVID, you will see many of the people are suffering from, inflammations, and they are going for varied kind of treatments. So how do we approach inflammation as such? One is the treatment which is already available. There are steroids, antibiotics, but what else can we do? So there are I had taken the paper of two scientists who have worked thoroughly in the field of on bioactive components, on phytochemicals, to look into what are the phytochemicals in which we can still research. We can still do a combination of them and bring more and more effective treatment for this particular disorder, which is bound to happen with aging. Yeah. So if we see its frequency increases with aging and approaches 40% in those over the age of 70. So if we see, it's a progressive disorder of joint, and in addition to damage, there is a remodeling of the bone, osteophyte formation, there is weakening of periarticular muscles, and in some cases there is also
Madam, you are muted somehow.
oh, sorry.
Oh, okay.
So okay, so the most common joints, was so I just wanted to tell that there are different joints in the body. So mostly hip and knee joints, the joints of the foot, the most common joints involved. Yeah, you will see that there is nip, there is hip, there is intraphalangeal joints and distal interphalangeal joints, which are being affected, involved in osteoarthritis. So I just wanted to give you a brief just before we move into that. So if you see, so the articular cartilage, it is a slippery tissue here, yeah, that covers the ends of the bones in the joint. And healthy cartilage, it allows the bones to build over each other, and it helps to absorb the shock of movement. Yeah. So in osteoarthritis, what happens? The top layer of the cartilage breaks down, and it wears away. Rubbing of bones under the cartilage takes place, due to which there is a pain, swelling, and loss of motion of joints. So over the time, joints may lose their normal shape, and there may be also growth of bony spurs on the edge of the joint, which causes more pain and damage. So, you can see like in normal bone, the normal muscle, whereas in osteoarthritis, there are weakened ligaments and muscles. Yeah, and there is destroyed cartilage in this case. So in osteoarthritis, there is progressive destruction of articular cartilage, and also there is involvement of synovium. So the fluid which is present inside the bone, which is synovial fluid, if there is excess damage due to trauma, due to it can be either, so it is characterized by like there is joint pain, there is tenderness, there is decreased range of motion, there is weakness, joint disability. Disability as such, you will see that the person starts limping, difficulty in walking. Yeah, so it can be primary osteoarthritis or secondary. So primary is like it can be localized, generalized, or secondary arthritis. It may be due to some other infection. Maybe if that person has autoimmune disorder, then due to rheumatoid arthritis, then there can be there can be because of post surgery. It can be due to trauma also. Sometimes that impact is so high that the person is under a trauma that it takes time to come out of it. Yeah. So it is one of the core, it becomes one of the core reasons of disability. So nearly everyone who lives long is affected by osteoarthritis at any one point in their life. And that's why, we have so many oils, especially from like Boswellia serrata. We use piper, we use aloe vera, we use a mixture made in ghee, made in, for in traditional therapies, we use that so that, it gives relief from that pain and inflammation at that time. So, as such, in this scenario, like 50% are those who are over the age of 65, and 85% are those who are over 75 age. And the overall grades of severity, it is seen like 0, 1, 2, 3, 4, like that. We grade osteoarthritis. There is it depends upon the kind of symptoms, the kind of when we go for MRI and other kind of tests, then we are able to see, what is, are there appearance of osteophytes, what is the joint space loss, sclerosis, and cysts. So the WHO adopted these criteria, when as the standard for epidemiological studies of osteoarthritis, like age, gender, ethnic group, specific joints involved, in method of diagnosis. So there is like hip, knee, and hand. Mostly, we say hip, knee, and hand for osteoarthritis. So between, you will see the percentage varies, and it enhances with age, whether it is hip or knee or hand osteoarthritis. It actually occurs more with age, and men are more affected after the age of 50, and women are affected after the age of 60. Yeah. In genetics also, they have seen, like in white population and Asian population, what is the prevalence that you can see. This is all the data. So diet is also very important. Like when we have osteoarthritis or arthritic-like symptoms, then you must try and use cold pressed oil. Take everything from home. Avoid all the junk foods. Yeah, avoid anything which can trigger it, like they say anything which is very salty, or if it is like to avoid a lot of sugar, to avoid a lot of soreness, like that. So it's like homemade food is always best. Yeah. So obesity is also one of the factor, and increased body weight is strongly associated with hip, knee, and hand osteoarthritis, and it often precedes osteoarthritis and contributes to its development. And with every additional, weight, though I'm also, I have to work on my weight. So the risk of developing osteoarthritis, it increases about 10% with each additional kilogram of weight. And in obese person without osteoarthritis, weight loss of even 5 kg decreases the risk of future knee osteoarthritis by 1/2. So we have to work. So many scientists have been working to see, new candidates from medicinal plant species and the natural products derived from them for regulating osteoarthritis. So if you see if you see the list of medicinal plants and natural products which have shown the effect on the pathophysiology of osteoarthritis, there are a range of medicinal plants which have been used and researched by all these scientists. So I've taken the reference of all of them. We have taken the paper of all of them, like Econitum is there, Bahunia is there, Boswellia serrata, Clematis, then you have Panax, then Philodendron, you have got Vidania somnifera, willow bark, gingiber office analysis, and then when you go for single compound, then you have mostly flavonoids.
Sorry to disturb, madam, but your voice is breaking.
Okay.
Slightly, it is some problem.
Sir, is it okay like this then?
Yeah, it seems to be fine. If maybe
now I cannot hear you.
Headphone or something.
Just 1 second. Oh, sorry. Sir, what I'll do is I will not speak from the Bluetooth. I will just cut my.
Yeah, directly you can. Okay.
Yeah. Just give me 1 second then, sir.
Okay, madam.
I'm sorry for this then.
So am I audible now?
Yeah, now it is perfectly clear.
I'm audible now, sir?
Yes, madam. Very clear.
Now I'm clear?
Yes.
Okay. So I was just telling, like, if we look into the, single compounds, so I will, if you want, I'll switch off my video also so that my voice is more clear. So if you see, there are single compounds like apigenin. So they're mostly flavonoids, they're terpenoids. You have phenolic compounds which have shown activity against inflammation. They have been used as anti-inflammatory agent, like the polysaccharides. They cause chondrocytes to express type 2 collagen, and they induce the expression of all this CDK6 and cyclin D1. They boost the chondrocytes cell cycle and growth, and thus they act as anti-inflammatory agents in and they are isolated from acantharis. So in Econitum carmelia, they have also shown inhibitory efficacy against the decline in bone density and cartilage deterioration. Okay, and when we come to medicinal plant, the total extract of Arnica montana, it has demonstrated anti-inflammatory effect in a rat model of collagen-induced arthritis, and it has decreased the level of IL-16, nitrous oxide, and IL-12. Additionally, the extract also has antioxidant property. When we look into astaxanthin, which is a carotenoid, it has also shown anti-inflammatory and antioxidant benefit on cartilage. It has prevented metallomatrix proteinase, including MMP3, and from expressing, because then they will inhibit, cartilage formation. So that's why this also has shown anti-inflammatory effect. Then apigenin, it is an anti-inflammatory flavonoid which has found to decrease the expression of MMPs, because if MMPs enhance, then it will lead to more inflammation. So additionally, apigenin intra-articular injection, it reduced the protein synthesis MMP3 in vivo and the rat knee joint also. This is a Apis mellifera bee venom. it has also shown to reduce the expression of metallomatrix proteins, which are activated, and they prevented the activation of TNF alpha. And astraglandin, it's also a flavonoid compound with anti-inflammatory and antioxidant properties. And this also has actually stimulated activation of NF K beta and MAPK in chondrocytes of osteoarthritis patients. Then another anti-inflammatory compound is acubin, which has inhibited the inflammatory response by blocking the phosphorylation and degradation and translocation of NF kappa B P65, and it is derived from variety of medicinal plants including Euphormia eulomidis. So we can use all these medicinal plants and the natural products. We can also extract them from different sources and utilize them for formulating new formulations for osteoarthritis as such. So we have seen that, biocillin, which is a natural substance derived from S. biocillinosis, it also inhibits the expression in human chondrocytes like MMP3 and MMP 13 and 3, because MMP is also an indicator of enhanced inflammation. So if you can inhibit the expression of MMPs, then it also indicates that it is a good anti-inflammatory agent. Then it has also been shown that polysaccharides which are extracted from Bahunia, they promote the transition of the G1S stage of the cell cycle, and they boost the proliferation of chondrocytes. So we all know that chondrocytes are essential for to prevent osteoarthritis. They play an important role. Then according to studies, bavacin, which is a phytoestrogen, it is found in medicinal plants Soralia corifolia. It also prevents cartilage deterioration and also by inhibiting nuclear translocation. Then berberin, we know from berberis, and in a human chondrosarcoma cell line, berberin, which is an anti-inflammatory natural substances which is produced from rhizoma, has been shown to inhibit cartilage breakdown and reduce nuclear factor signaling pathways. And it also showed a potential chondroprotective impact by reducing chondrocyte death. So all of them have been proven, all these medicinal plants. Another scientist had isolated almost, he has made a mixture of almost 43 herbs, which is found to be so effective against osteoarthritis. So betulin is another naturally occurring anti-inflammatory substance which is isolated from betulia, which increase type 2 collagen gene expression. And then we have biochanin A, which is isolated from red clover, which is found in like Nepal. It is called Lal Tipatia, and it has been shown to interfere with the signaling pathway in chondrocytes, and it also prevents the expression of MMPs and restoring the expression of TIMP. So Boswellia serrata oil we use in general, we use a lot. So it has demonstrated, if you see the whole plant extract has demonstrated antioxidative and anti-inflammatory properties in the animal model. And patients with knee osteoarthritis in a clinical trial, when they received treatment with the extract, it reduced pain and improved the functionality. So if you come to catechins, there is epigallocate chin 3 gallate, a representative catechin, which has been reported to exert chondroprotective activity, and they are the primary polyphenolic compounds which are found in tea, and they have potential anti-arthritic effects. Then there is cholesterol, which is a natural substances isolated from the root extract of thunder god vine, which is Tryptigum wilfordii. It inhibits heat shock proteins, and it has also shown a suppressive impact on stimulated production of MMP13 and MMP3 in primary human osteoarthritic chondrocytes. Then we have climatic kinensis. So we can all work on these medicinal plants, or we can choose one or two in which we want to further do our research, for bringing out. We have to research it again and again. So we all know, why chondrocytes are so important, and chondrocyte is coming again and again because they maintain the extracellular margin, a matrix, and produce the cartilage matrix. Yeah, and they are surrounded by collagenous fibers, and they release substances to make cartilage strong yet flexible. In general, they are found within intervertebral disc and in any form of articular cartilage. And that's why, we are always focusing the scientists all over who have been working on single entity, or whether a particular flavonoid or a saponin fraction, like in this they have isolated from clematis chanensis, which demonstrated an inhibitory effect against cartilage damage and destruction of the joint in an animal model of osteoarthritis, which is created by the intra-articular injection of monosodium iodoacetate. So this was accomplished by suppressing the breakdown of extracellular matrix and chondrocyte injury. So crocin, so by blocking the NF K beta signaling pathway in articular chondrocytes, the natural substance crocin, which is isolated from crocus, it is found to prevent both the expression of MMP13, MMP3 and MMP11, as well as degradation of cartilage in vivo. And then we have the famous, curcumin, which has been researched for so many things, and we know in general also we apply it, and it is a proven anti-inflammatory agent. And when it is used on an animal model on arthritis, so for preclinical studies we go up to animal modeling, though we have cell lines, but then we confirm it through animal modeling also, that when we have applied this curcumin to an animal model, like where arthritis was induced on the animal model, the substance reduced joint inflammation. And in an in vivo arthritic model, a formulation of curcuma longa whole extract, it reduced joint inflammation and periarticular tissue damage by blocking the signaling pathway. So, where does India lack? Where does we all lack? Though we have such rich traditional knowledge, and we know, this extract will work for this, is the scientific basis. So that's why, all the scientists are now working, and government is also introducing funds in that direction to prove what is already proven, but to prove it in a scientific way that yes, this works on this particular, it acts like this in this pathway to show anti-inflammatory effect, or if it has antipyretic effect, or if it is working against, psoriasis, how it is working against cancer. So that scientific data is now being collected by all scientists all over the world. So including osteoarthritis, in which the traditional medicines are more effective than the allopathic drugs, because the allopathic drugs, they damage the liver. They have so many side effects. So we must continuously research. As researchers, we all have that responsibility also to continuously touch on the topics which actually affect the human life. So one is cancer, which is always ongoing, and people have really fine breakthroughs. Another is these degenerative diseases, which actually affect day to day, every person above the age of 50, and when they, even the small children are getting affected. So another one is delphinidin, an antioxidant, anthocyanidin, which is present in a variety of vegetables and fruits, and which inhibited the production of COX2 and PG2 in osteoarthritic chondrocytes, and it also inhibited NF-kappa B signaling, and thus it acts as an anti-inflammatory agent. And like another one is like if you take an aqueous extract of Euphomia ulmoides, which is normally found mostly in Japan and China, they have shown anti-osteoarthritic properties. Then there is fluoride acid. So by preventing hydrogen peroxide induced production of MMP3 and MMP1 in chondrocytes, ferulic acid, which is a naturally occurring substances, it has also shown anti-inflammatory effect. And this is one formulation which is developed in South Korea, which is a combination of 6 herbs, and which is there is glycine, there is Euphomia, there is acantharis, and intra-articular injection inhibited the formation of anti-type 2 collagen antibody, and it was tested on osteoarthritis on an animal model in which osteoarthritis was created by monosodium iodoacetate. It controlling the balance of cytokines and inflammatory mediators. Then, ginseng, so you get ginsenosides, and different biological effects were seen in ginsenosides that were isolated from Panax ginseng, and according to their reports which have been generated, they have a chondroprotective action. So anything which protects the chondrocytes, which actually helps you for the smooth movement of your different joints. So chondrocytes, anything which will prevent the damage of cartilage, the damage of chondrocytes, which will inhibit the expression of MMP proteins. So that is very important. So another medicinal plant which is called as devil's claw, a folk remedy. It has been used to treat osteoarthritis and musculoskeletal degenerative illness. And in this, the whole extract is taken, which inhibits again the activity of MMPs and elastase, as well as the production of inflammatory mediators. So in a clinical experiment which was done, it had shown that various HP extract had beneficial effects on individuals with hip and knee osteoarthritis who were experiencing pain and very limited movement. Then there is another natural chemical which is derived from, magnolia. It is found in Japan, and that is called honikiol. Then it's a polyphenol. Then we have acarin, which is a substance produced from epidium, and it has shown to suppress expression of MMP3, and additionally it increase the production of extracellular matrix, and it has chondroprotective properties. Then another was luteolin, which is a flavonoid compound which is derived from L-flowers, and it also mostly, they inhibited the gene expression in MMP 13 or MMP1 or ADAMTS 5. Those proteins were affected. So if you see MMP plays a very important role. If it is not expressed, you should prevent the expression in osteoarthritis. So this matrix metalloproteinases, they're also called as matrix metallopeptidases or matricins. They are calcium dependent, and they play an important role in tissue remodeling during various physiological processes, and during if there's any change in the body, yeah, you will see the expression will also change for MMP. If there's any change, if there's a tumor, if you are undergoing any kind of change inside the body. So it is especially like MMP8, it is a tumor suppressor gene. So like that, like it plays a very important role. Then we have a substance which is found in morinda, which is known as monotropin, and it inhibits the chondrocyte MMP13 and expression. And then there is a flavonoid morin, which has shown anti-inflammatory, antioxidant, and also anti-cancer properties. So if you see, we also have philodendron amorens, which has immunostimulatory and anti-inflammatory effects. And there is another ingredient which reduces the production of 13, which is pinacombrin. Then the use of herbal remedies in treatment and prevention of osteoarthritis, it has drawn more attention in recent years. And the herbal ingredients that have antioxidant, anti-inflammatory, or cartilage protective properties have also been successfully employed to treat osteoarthritis. And cheminformatics tools were used to forecast the drug-like properties. And this is the latest research done by one of the scientists in China, where they have used cheminformatics tools to forecast drug-like properties and pharmacokinetic parameters in order to serve as the foundation for the subsequent studies and advancements. So they have, they had utilized PubMed and Google Scholar to gather all the information for the treatment of osteoarthritis by bioactive components from herbal medicines. And 43 bioactive components were identified to have anti-osteoarthritis properties, which included 11 terpenoids, 10 flavonoids, alkaloids, phenols, quinones, coumarins, lignan, steroid, and furan. And the cytospace program was created, which created the network of osteoarthritis pathogenesis and also chemical targets. So this is the latest research which is done in 2022, where they have used cheminformatics tool, and a cytospace program, SCAPE program was created, which created the network of osteoarthritis pathogenesis and chemical target. And the primary feature of osteoarthritis we know is joint deterioration, which is accompanied by alterations in relevant circulatory system markers. So the pathophysiology of osteoarthritis, it involved numerous pathogenic processes like inflammation is there, apoptosis is there, oxidative stress, and others. So they have utilized all these mediums like the PubMed, the latest research. They gathered all the information till date, whatever was done. So there were 43 bioactive components. Yeah. So they created like the main pathogenesis and lesion sites of osteoarthritis. The blue circular mode, if you see, is the pathogenesis, and the red diamond-like mode, they are the cartilage damage. The green colored diamond mode, which you see, subchondral bone damage. And the blue diamond mode, yeah, it indicates synovial injury. Pink diamond node included circulatory system changes. And purple diamond mode, which is there, which includes muscle weakness and inflammation. So this is how they, with the help of cheminformatics, they had a cyberscape in which they had pathogenesis. They took various, markers which indicated, any one of them will indicate that the person is suffering from osteoarthritis and how the herbs are going to treat. So these bioactive substances like resveratrol, curcumin, what we have studied, they primarily target the circulatory system and damage joints. So, they have taken everything like circulatory system changes, yeah, and subchondral bone damage, then muscle weakness and inflammation. So if you see the bioactive components, which are like resveratrol, curcumin, they primarily target the circulatory system and damage joints such as cartilage, synovium, for example, and they improve the pathogenesis of osteoarthritis. And these substances, they primarily exert anti-inflammatory, anti-apoptotic, and antioxidative stress effects, yeah, through interleukin, nuclear factor B, and matrix metalloproteinase. Okay. So another view, the overview of targets and effects of bioactive components with anti-osteoarthritis activities. So there are different colors which are given, like green. The green color in this indicates herbal compounds. The yellow circular node indicates the targets. The red indicates the anti-inflammatory effect. So, taking 43 herbs and taking the bioactive component, they were able to do this kind of pathogenesis to come out with, an effective formulation for osteoarthritis. Then the green rectangular node in this indicates anti-apoptosis. The blue indicates antioxidation. The pink, which you see here, is anti-catabolic. The purple is anti-angiogenesis. Orange is anti-osteoclastogenesis. Then individually, if you take, the overview of anti-osteoarthritis activities of, like, A is resveratrol, B is for curcumin, then for berberin, imodin, yeah, and again, they saw the effect like this. So they were herbal compounds, and the dose was, the effective dose was decided both in vivo and in vitro. So like for, if we say berberin, so in vivo that was 10 to 50 milligram per rat. Yeah. And like the dose started from around 7 microgram to 50 milligrams. So they enhanced the dose and saw the effect, yeah, in vivo and both in vitro. Like for curcumin, it was taken 50 to 100 milligram per kg per mouse. So we know it's according to body weight, like that. So like if we take resveratrol, which is very which is widely used, they had taken 45 milligram per kg for mouse. In rabbit, it was tested for 10 to 50 micrometer, then micromolar per kg. Then it was like 30 to 120 milligram per kg for rabbit. It was tested. So synovial joints, like they have seen the effects of bioactive elements on osteoarthritis cartilage. And yeah. So if we go on, like we have seen that curcumin, berberin, tetramethylpyrazine, ferulic acid, chiconin, gensoicide, then we have crocin, piperine, butene, yeah, all of them inhibited the expression of metallomatrix proteinase via numerous pathways, and consisting of interleukin 1 beta signaling, nuclear factor signaling, yeah, and nitrous oxide signaling, etc. And the inhibition of cartilage with the aid of using resveratrol, curcumin can be additionally, we can associate with the law of autophagy. Activation may also lessen the severity of osteoarthritis. And oxidative stress is another key factor in the development of osteoarthritis. And if you see, cartilage exhibits dysregulation of a number of oxidative stress marker, including nitrous oxide. And there are several compounds that can prevent the abnormal expression of these indices, which includes resveratrol, piperine, butene, yeah. And it is so we all know that oxidative stress, it is brought about by mitochondrial failure in chondrocytes, which is linked to osteoarthritis. So like all these indicators, so the onset of osteoarthritis is also linked to abnormal angiogenesis. So by interfering with a number of targets, including vascular endothelial growth factor, nerve growth factor, several herbal substances inhibit aberrant angiogenesis. So when you know the markers, the points like the inflammation, the angiogenesis, atopsis, oxidative stress. So the target is being set based on natural products on these particular components. So bioactive components, they affect the subchondral bone also in osteoarthritis. And because along with cartilage, the subchondral bone lesions, they are also degenerative alteration that characterize osteoarthritis. So they also affect that. And then the synovial fluid, which is present inside bone, it feeds the cartilage with nutrition, and it shields the adjacent musculoskeletal injuries and joint structure. So osteoarthritis is a typical synovial degenerative condition, and both osteoarthritis symptoms and progressions are both impacted by synovitis. So the process because of which there inflammation occurs in osteoarthritis, which also promotes synovial growth, it can be stopped by anti-proliferative properties of curcumin and icariin. So all the scientists who had worked in the past, they have again validated this with the root, whether it is targeting the effects or it is targeting the targets which are there, whether it is synovial fluid, inflammation, oxidative stress, the trauma, all we have our the bioactive components are there. And this is the latest research which is going on to see whether the different aspects. So one of them is also that the circulatory system effects of bioactive components in osteoarthritis. So circulatory pathology and osteoarthritis, they have a very close relationship. So and numerous herbal substances have been shown to be able to reverse some degenerative processes in osteoarthritis model serum, and they may be regulated by such bioactives like quercetin and artesunate. So if you see, when you are looking into oxidative stress, there curcumin is there, but when you are looking into circulatory pathology, then you look into other bioactive component like quercetin and artesunate. And it also affects, there is muscle weakness and inflammation. Yeah. So by reducing inflammation, crocin, it reduces the symptoms. Yeah. So we can use for muscular weakness, the bioactive component which can be used is crocin. Yeah. So when we know what are the symptoms, accordingly we can, with the help of our team, we can develop, such kind of candidates for future studies, and which can actually have good anti-osteoarthritis properties. They have it, we have to just prove it. Yeah. So chem informatic tool plays a very important role. And so I think I'm running out of time, but I would just like to say that, they have outlined 43 herbal substances in that, in the particular review that the scientists have done. I've cited their reference, and more than half of these substances have favorable drug-like characteristics, and they may warrant additional research and development. And they are mainly found in plants belonging to Leguminosae and Polygonaceae families. And so we can look more into those families for carrying out our research. And the pharmacokinetic behavior of these components still needs further refined through cheminformatic studies, and which actually is helpful in improving their drug-like qualities. So as pharmacists, many of the students can work on the pharmacokinetic behavior of these components. and currently there has been some advancement. However, the use of herbal substances is still very insufficient when compared to treating other form of arthritis. So inflammation, apoptosis, oxidative stress, and other pathogenic factors, they are shared both by osteoarthritis and rheumatoid arthritis. And so that's why, further studies can expand the potential of anti-rheumatoid arthritis medicines in the treatment of osteoarthritis. And we need several research methodologies like systems pharmacology, kinmemodemics, and biolabeled research methodologies. They can be employed in future to accelerate, and they are being employed also to accelerate the investigation of the mechanism and the chemical foundation of herbs in the treatment of osteoarthritis. So I will just take 1 minute, like we have also done a lot of innovations in our lab. I'll just make you go through, like we have developed this is all based on the traditional knowledge we have developed. So all these references have been used for this presentation. So it's a long list of references. So I thank you so all for your kind attention. I'll just open my video also. So it's a vast topic and it needs constant research, and we can just focus on 1 or 2 medicinal plants also. But recently, like a cheminformatics tool had been used on 43 herbs. So I wanted to just go through it one by one and also show how they have used different indicators, different markers to come out with the chemical basis of the herbs, so that we can utilize it further. Yeah. So thank you, sir. Thank you, everyone, for your kind attention.
Thank you, madam. It was a nice presentation. Good morning, madam. Kanchan Kohli madam has also joined.
Yes, good morning, ma'am. Kanchan Kohli, ma'am. You're muted, ma'am.
How are you?
I'm fine, ma'am. How are you, ma'am?
I'm also fine. It's good lecture, and we gained so many things from your lecture, that what molecules you are touching and how you are doing it. So very nice presentation.
Thank you, ma'am. Thank you so much, ma'am.
Very informative.
Take up questions, or we will reserve questions to the end and move forward for next speaker.
Yes, sir.
Yes, sir, we can move, I think.
Yeah.
Thank you very much, Dr. Harsha Karkwal, for your presentation and giving eyesights into various aspects of approach towards the target, as well as drug approach to towards the targets in osteoarthritis. Now I take opportunity to welcome Dr. Kanchan Kohli madam to this webinar. On behalf of the management, staff and students, I welcome you, madam, to this webinar. It's a pleasure to introduce madam to our webinar. Dr. Kanchan Kohli is come from PhD and professor in Department of Pharmaceutical School of Pharmaceutical Education and Research from Jamia Hamdard University, and that is top 1 in NIRF ranking. She had a very long exposure of teaching as well as research, and she received PG Research Award UGC in 2002 for research topic novel mucoadhesive formulations of periodontal diseases for a period of 2 years. Received Best Project Award from the hands of the Vice President of India on the Technology Day in 2002-03, organized by DST as well as the All India Council for Technical Education. He has been very active in delivering number of plenary lectures and resource person for various professional bodies, etc. And more importantly, she is nominated as a member of expert committee group herbal products by Indian Pharmacopoeial Commission, Ministry of Health and Family Welfare. Her name will be there in all pharmacopoeias as in the preliminary pages. She won IPA ACG SciTech Innovation Award for basic innovative developments on solid dosage forms in 69th Indian Pharmaceutical Congress at Chitkara University. She had patents, 15 patents to her credit, and 2 patents have been accepted from ICMR, and he has US patents, and he has guided M.Pharm dissertations 45, and she has awarded PhD to her students number 25, and she is a very active a researcher in the pharmaceutical sciences and coming from Jamia Hamdard University. In fact, if you browse her name in Google search, you will get lot number of publications, etc. But she is also modest in telling the truth. Now it is time for us to listen from her mouth what are the research innovations in that she has taken, and innovations related to nanoparticles of herbal drugs. This is aptly suited for the webinar topic. With these few remarks and introduction, I welcome once again Dr. Kanchan Kohli to this webinar. I handed over the mic to Dr. Kanchan Kohli. In fact, Muhammad Ali was my colleague in RRL Jammu. We were
very
there
nice.
for 4 years
Okay.
in plant chemistry, anti-amoeboic chemistry.
Okay, very nice, very nice. I worked
Here I would
for
like to
my
add one thing,
M.Pharm under-
madam. With
Yeah.
all that, whatever CVS sir has read from your biodata, still, after all these achievements, you are such a down-to-earth, simple person.
Oh, thank you so much. Thank you so
Definitely,
much.
I am most happy to have you at least online in our institute.
Thank you so much, Dr. Kulkarni. Thank you. And thank you, Dr. Pani Kumar, giving my introduction. And it's very nice to hear from you that you have worked with RRL Jammu. And I mean, with such a vast research experience, I'm really very happy to see you and share my, I mean, the views, whatever things I'm having in my mind, I'm sharing on the platform, and very happy. And thanks to Dr. Kulkarni and Dr. Pani Kumar to invite me or to share this platform with I mean, your audience and other dignitaries. Thank you so much. So with this, I'm just starting with my today's lecture, that is we can say innovations related to nanoparticles of herbal drugs. So as
I will
we
start
all,
sharing the screen, madam.
yeah, please. So this is on the topic, that's the innovations related to nanoparticles of herbal drugs. I'm sorry to welcome all the participants who are joined this presentation, my presentation. Thanks to all. So this is the topic, innovations related to nanoparticles of herbal drugs. So today, what problem we are facing with the herbal drugs is that bioavailability issue is there, solubility issue is there, and maybe the drugs which we are, or the herbal particles which we are consuming, they may not be going to the reaching to the blood at all. And what innovations we can do with that? And frankly speaking, today's work, or what I'm pursuing my work, that is converting this or changing this conventional system to modern system. So with changing this modern system, what I'm achieving, that one thing is there, that whatever minimize we are side effects are there, and moreover we are increasing the solubility and the bioavailability of the product. So this is the main, the innovation we can say, but still many problems are there. And with that, with herbal drugs and with those problems, we are going ahead, and maybe the day will come, the many product will be from the India, which will be exported to other Western countries. Very soon the day will come, because our scientists are working very hard for this field. So next slide, please. So novel drug delivery system is advantages in delivering the herbal drug at the site of. This is the very important thing which we can deliver with the NDDS, that at the site of the action, when we are giving the drug at the site of the action, then definitely we will be minimizing the toxic effect and we are increasing the bioavailability of the product. So today I've, as mentioned, that today the most problem with the herbal drug is that whatever constituents we are taking it, or the phyto constituents even we are taking it, this stability issue is there, and along with the stability, the bioavailability issue is there. So what our aim is to improve the bioavailability of this phyto constituent so that they can be more useful for the community. So second point is, in NDDS, the control of distribution of drug is achieved just by incorporating the drug in carrier system, or in changing the structure of the drug at molecular level, which will come afterwards. So herbal drugs are becoming more popular in the modern world for their application to curb variety of disease with less toxic effects. Of course, the toxic effects are there. I mean, this myth that we don't have any toxic effect with the herbal drugs, it's just myth, but still, I mean, the toxic effects are there. And when we go through the literature, we found many, I mean, the toxic effects are there with the herbs. So what is our aim is to how we can reduce the certain toxic effects. And when we are moving towards NDDS, definitely these toxic effects could be reduced and we will be having the better therapeutic effect. Next slide, please. The novel carrier. Next slide, please. So firstly, it should deliver the drug at a rate directed by the needs of the body. So what our novel preparation fulfill the requirement? So first requirement is deliver the drug at a rate. Okay. So second requirement is it should channel the active entity of herbal drugs to the site of action, and it should take at the site of the action, because you know what happens when the bioavailability issue is there, when the absorption issue is there, then what happens, the drug or the molecules just stick at a time. Like when we have seen the curcumin results, if the curcumin was not in the nano form, or we have not changed the technology, that then we have seen all the material or the drug was stick at one part when we have done the gamma scintigraphy studies. And afterwards, when we have converted to the nano formulation, then we have seen the drug is moving towards all vital organs. So this is the achievement which we can get when we are converting to the novel drug delivery system. So next slide, please. What are the advantages of novel drug delivery system? That it enhancement of the solubility. When we are increasing the solubility, definitely the bioavailability will be increased, and when bioavailability will be increased, the dose of the drug could be reduced. Then dose of the drug will be reduced, the toxicity level will be reduced, and then enhancement of pharmacological activity, enhancement of stability. Today the much issue with the phytoconstituent is there. Whatever even previously madam has told, whatever the drug constituents are there, they are quite unstable. I mean, they are not stable at, say, even sometime at 45 degrees centigrade, 55 degrees centigrade. Then what's our aim as a pharmacist? That we should increase the stability of the product. So how we can improve the stability of the product? There's the big question mark, and we have to all work in this direction, because our most of the herbal constituents, they are unstable. So sustained delivery, we should achieve in the sustained delivery. Production from chemical, of course, this is again to related with the stability issue, that's the chemical and physical degradation. And sometime we have seen our most of the phytoconstituents, they are phyto degraded.
So then we have to save them. How we can save them? How we can improve the stability of the product? So targeting of drugs in particular tissues, organs. So these are the all the advantages of your herbal when we are converting to a new drug delivery system. But of course the limitations are there, because in the herbal drugs we are having the polyherbal formulations, and working with the polyherbal formulation is really difficult. We have to see the biomarker of that each of the herbs, and then we can standardize it, optimize it, and then ultimately the stability issues could be there. So many problems are there with the herbal drugs, but still if we start working with one drug, definitely, I mean, we can go ahead with and we can just bypass the word, and we can go better in the then export of our formulations in a better way. And then India can be the number one in herbal drug productions. So next slide, please. So herbal drugs, herbal formulation means a dosage form that's consisting of one or more herbs or processed herbs in specific quantities. Processed herbs means we will be having some kind of the phytoconstituents, we'll be having the kind of the extracts. Sometimes, you know, the extracts can be of the two types. One is the water soluble extract, other that's the lipophilic, and other is the hydrophilic. So where we have to see the constituents in which, this is the again the big problem with the herbal extract, you know, we really don't know. I mean, our constituent has gone to which side, whether it is the lipophilic side or whether it is the hydrophilic side. Means lot of the work is required. Of course we can conclude that where our activity will go, but we have to do the lot of the analytic work for that reason. And for every step we have to go for these HPLC studies, and we have to adopt other analytic methods for that reason, and could be very expensive for us also, because in synthetic drugs you will be finding, when you are going for the literature, you will be getting all kind of the data, which is difficult in the case of the herbal drugs, because in herbal drugs this standardization has not been done in a proper way. But nowadays I'm finding the papers, people are coming with the kind of the combination of the herbal drug with the synthetic drug, and combination of 2 drugs, 3 drugs. People are doing the research work, and the day will not be very far when we'll be having kind of the products from the herbal formulations. So advantages of herbal drugs, then we are having the low risk of side effects, lower cost, widespread availability. When we talk about the lower cost, presently I don't find that we can have the lower cost of the product, because, you know, we have to carry out lot of the standardization for the herbal drugs. Even, you know, when we are getting the raw materials, we don't know, maybe some kind of the fungus could be there, and with fungus might have they have grinded, the suppliers have grinded those fungus also, or insecticides, percentage of the insecticides, percentage of the we can say metals in the that's the major problem. The major problems with the herbal drug is that we can have the metal content in that formulations. So when we are analyzing it and when we are standardizing, then it requires lot of patience also, and lot of finances also. So today I am not convinced by this, the lower cost. Of course, once we have optimized the formulation, when we have standardized, and when we have got the good extracts to work, then only we can say it could be lower cost. But presently it's not that. Next slide, please. So the major problems with herbal drugs is poor solubility, that's characterizing in poor oral bioavailability, for which we are going for the innovations, and the instability, which we have discussed earlier also. So in stability, like environmental conditions, presence of light, heat, and oxygen. So this gives challenge for formulation approaches to develop better pharmaceutical formulations with improved solubility, stability, and enhanced systemic bioavailability characteristics. So now we'll see what the novel approaches could be adopted for the development and for the formulation of the herbal drugs. Next slide, please. So the novel approaches, of course, the nano formulations are there, and we can have the micron also. So directly we can say micron to nano formulations when we are going, then much of the problems we can solve it, like especially the solubility and the bioavailability. Of course, for the stability, we have to see what the stabilizer we can use in a better way to protect our phytoconstituents, active phytoconstituents, how we can protect them. So the novel approaches, I'm going hurriedly, it's going for the nanoparticles, solid lipid nanoparticles. Of course, the nanoparticles, we all know now what the nanoparticles are there. So micron to nano, so they can be in the form of the nanocapsules, nanospheres, nanoclusters, nanotubes, nanopowders, nanowires, etc. But again, we have to standardize our products. Nanoparticles of herbal drugs like curcumin, like lycopene have already been formulated, and they have formulated in our lab also. And I'll show you the data, how we have improved the solubility and bioavailability of the product. Just by knowing the thermodynamic studies and kinetic studies, we have concluded it. So in nanoparticles, we will be having the solid lipid nanoparticles. So as the name indicates, solid lipid nanoparticles, nanostructured lipid carriers, whatever the lipid carriers are there, we are conjugating our products, herbal drugs, with the lipids. So it depends all from, next slide, please. So we are having the solid lipid nanoparticles, and we are having the nanostructured lipid particles, lipid drugs conjugate nanoparticles. Now, every system is having advantages and disadvantages. Like in the case of the SLN, we have say the unpredictable gelation tendency is there. So particle size growth, poor drug loading, then difficult to load hydrophilic molecule and relatively high water content. So then we can switch over to the NLC. In the difference between SLN and NLC, out of these, I find the NLC are better. Here the loading is much better as compared to the SLNs, we are getting the kind of the dispersion, but here NLC maybe we are getting because the liquid lipids are, so NLC is the combination of the solid and the liquid lipids. And in case of the SLN, normally we are using the lipids, solids one, and here we will be getting the kind of the clear formulation or the transparent preparation as compared to SLNs. So whenever we are having, and in both cases we are finding the loading of the drug is very low, whether it is SLNs or whether it is NLC or whether LDC. So the question comes when the loading is poor. So in herbal drugs, you are finding the quantity of the drug or the quantity of the extract is too much, like maybe it is 1 gram to 2 grams. So in case of that 1 gram to 2 gram, these our methods will not work. Like we will not work. NLC, LDC, nano, sometimes maybe nanocrystals could be better off, and we will be having the other system kind of the nano emulsion, or better is self-emulsifying drug delivery system, which will come afterwards. So we will be having the nanocrystals. Next slide, please. So nanocrystals. Next slide, please. Next slide, please. So we will be having the nanocrystals, nano emulsions. then nano emulsions. Then we will be having, next slide, please. So in nano emulsions, we are normally having kind of the, as we know, emulsion is always, we can say, you're the immiscible liquids, but here we are having the two kind of the surfactants. One is the surfactant, main surfactant, and other is the co-surfactant. So this is kind of the co-surfactant delivery, in which we will be having the thermodynamic stable. But the main problem again comes with the nano emulsion, that's the stability issues. But nowadays the formulators have seen this problem, and they are we are having now the kind of the excipients that's been formulated by various we can say the big companies, and they have come into the manufacturing of the API, you are the excipients only. And this is a big market of the excipients nowadays. And maybe you people have visited expo or like that, in which many suppliers are there only with the excipients. So the big demand of the nowadays excipient is there. Even you can, I mean, the young generation, if you can start with your business with the supply of the excipients, you can earn a lot, because newer and newer excipients are coming to the market, and much demand is there. And only thing is, we have to show the better achievements in that way. Maybe in better achievements like the solubility issues or like the bioavailability issues. So the next system, we are having the self-emulsifying drug delivery system. In this system, we have seen the much problems which we are facing with the SLN, NLC, LDC. That's where the loading of the drug is much less. Here we have seen in this case, the loading could be improved in case of self-emulsifying drug delivery system. For example, you are having the 500 mg of the oil, 500 mg of the drug, which has to be delivered. So that's near, we can have the kind of the loading, say 500 mg of the material will be there, and maybe you will be adding 200-300 your ml of the excipients into it. So the total volume may come 800 ml, for example. So the 800 ml of the preparation, you can very well have the soft gel capsules, or nowadays we have filled the preparation, that is the curcumin preparation, in hard gelatin capsule. The reason was there, when we have added, excipients, the latest excipients, we got it leaching in the soft gelatin capsule because of the gelatin interaction. So hard gelatin capsules worked much better, and today we have filled the liquid formulation in the hard gelatin capsules. And for that, of course, the the different kind of the filling machine is there. Very different, not very different. I mean, only the one attachment is required, where we will be having the double sealing of the capsule. Otherwise, the liquid will come out from the capsule. Like in the case of the solid, we are having just sealing, but here we are having the double sealing. So these are the SADs are your isotropic mixture of oil, surfactants, solvents, and co-solvent surfactants, and can be used for the design of formulation. As the name indicates, self-emulsifying. Initially, what the formulation will be there, like it's a combination of the surfactant, oils, and solvents. Maybe it means it is just the lipophilic system is there. But when it will be reaching to the system, there it with the amalgamation of water content, it will convert to the emulsion, and that's why it is called as the self-emulsifying drug delivery system. And that emulsion which we will be getting it, that's in the nano form. So similarly, we are having the SMADs. In case it's SMADs, and the difference between SMAD is not much difference is there. There's a small particle size difference is there. But again, the major differences between SNADs and the SADs is there we are needing, in case of the SMADs, more quantity of the the oils or the surfactants is required as compared to the nano system. And in some drugs, nano system is better, and some drug, the micron system is better. We have to ultimately see the stability, where the stability of the product is there, whether it is SMARTs or whether it is SADs. So self nano emulsifying drug delivery system and self micro emulsifying delivery system, as I mentioned, not much difference is there, only kind of the little bit particle size difference is there, but the much difference is the quantity of the lipids which we are using. They are much higher in case of the SMARTs as compared to the SNADs, and therefore we have seen the SMARTs preparations, they are more stable as compared to the SNADs because of the much higher quantity of the lipid present over there. So this is the different. Then we are having the liposome preparations. So as you know, next slide, as you know, the liposomes preparations are coming very common to the market, and we do have in the our national and international market the liposome preparation. And you might have heard during the COVID time, we do have the two preparation of amphotericin, which was mainly given for the cause of black fungus, when the black immunity lowering was there, and the kind of the black fungus occurrence was there. During that time, you have seen the amphotericin preparation was used. So there the amphotericin preparation is the liposomic amphotericin preparation, whereas in market we had two preparations. One was the plain preparation of amphotericin, other was the liposomic preparation of the amphotericin. But our physicians were not satisfied with the plain preparation. They always wanted the liposomic preparation, and you might have heard in between the shortage of the amphotericin preparation was there. So this was the main reason, because formulators were not aware that much quantities of the amphotericin will be required, and suddenly all happens. We were not knowing what therapy should be given and what not, and you have seen all episodes were there. And during that time, why the shortage of the amphotericin liposomic preparation was there? Because only one or two manufacturers were there, and they couldn't supply in the sufficient quantity to meet the requirement of the public. So what happened then? Suddenly, to locate kind of people who are much aware of it, the liposomic preparation, that technique was not available with the manufacturer, and people were not aware of that. And to stabilize this liposome preparation, it takes time, and that time was not there. And whosoever manufacturer was there, that's why the shortage of the amphotericin liposome was there. And after that, I've heard that so many scientists are working for the liposomic preparations, but because as the awareness of the liposomes is there in the public, so even whether people are from scientific background or not, but they are now aware what the liposomic preparations are there. They know that they are much better than the plain formulations, and they will come when we will be having more and more formulations from the liposomes also. So again, to stabilize this liposome is difficult task, which our scientist or which our formulator has to take the initiation of that. Similarly, from the herbal drugs, we can have the liposomic preparation, and when we are going for the research papers, many people are working for the liposomes. But sometimes, when you are moving to the research papers, you don't know whether actually people have worked to stabilize those preparations or not. Simply they have worked and they have taken the size and they have shown it's converted to micro size. Or in liposomic preparation, it's not very essential that the preparation will be in the nano form. It could be micro form also. So the thing is, here we are also using these phospholipids, and with those phospholipids we are enhancing the solubility and the bioavailability of the drug is there. As you are finding the structure of this photo, you will be finding the, I mean, the absorption of the molecules will be there, and definitely, I mean, the solubility issues could be solved out. And today we are having the from the liposomic drugs, we are having the anti-cancer drugs like doxorubicin, which is available in the market. There's the Doxil and camptothecin, and mainly we can say the anti-cancer drugs are available today in the, and many more antibiotics drugs, this as given you the example of amphotericin. So they are available. Similarly, from the herbal drug, today we are having the silymarin. There's the liver flow micronized liposomes delivery, delayed release dual capsule technology is also being currently marketed. So these were the fewer concepts or the innovations which you can bring to the herbal drugs. And whenever you see you are going for the any system, you have to see the characteristics of the drug molecule, then accordingly you can start with your system. Many students or our scholars, they don't go for the literature survey properly, and they start working it, and in between they land to the problem. So first thing is the literature survey should be proper. You should spend 2 months, 2.5 months just to find out the literature survey. Then your things will be clear in your mind. And whenever you start working with your research work, first properly go to the literature survey, national level, international level, Science Direct, all the sites you should see correctly, and then you can decide that what system you have to follow and what way you have to go to improve the solubility and bioavailability issues of the herbal drugs. So today I mentioned we are having the 4 main issues with the herbal drugs. That is your solubility, of course, the bioavailability, stability. Stability is the main issue with the phytoconstituents, and as you know, now the drugs with phytoconstituents are mainly coming, and our DCGI office is giving the approval for phytoconstituents. Whereas if we have to go for the herbal formulation, we have to move through the Ayush. Okay, so this is the way we can have our license. Whenever we are going for the DCGI route, then we can go for the phytoconstituents with proper references, with at least 4 references. This is the initial requirement of the DCGI, and of course first your phase of the clinical trials. So now I'll show you, I mean, for which nanoparticles of the herbal drugs we have worked in our lab. I've started working kind of the long back in 2011 and 12 with these herbal drugs, only with the kind of the innovations, what innovations I can bring and how we can improve our factors which I have mentioned. Then of course, as I mentioned, that many limitations are there, but we have to see what are those limitations, how we can improve those limitations. Because sometimes what happens if the drug is having higher dose, when you are going for the nanomicron size, may of the drug of the dose will be much reduced, kind of you will be having the 1000 milligram with the drug, it may come to the 500 mg also, 400 mg also. So it all depends, and that factors we can conclude with the help of the pharmacokinetics and dynamic studies, and comparing, maybe that time, particularly initial stage, we can compare our studies with the marketed synthetic drugs, because when the available data is not there, so we can very well compare with the synthetic drugs, where we land, where is our motto, where we have reached. So after that, we can conclude our results that we have, maybe the day will come, we can replace our formulation with the synthetic drugs. So this should be the motto. So some of the our work that's related to nanoparticles of the herbal drugs. First, we have worked for the curcumin, and in curcumin, we have very worked for, we have improved the solubility, and we have prepared the snacks of this preparation. There's a self-nano-emulsifying drug delivery system. And when we have compared with the plain curcumin, of course, we can use the curcumin for many reasons. And before me, madam has also mentioned about the curcumin molecule, which I have heard. So we can have the many therapeutic uses of the curcumin. It could be cancer, arthrosis, diabetes, respiratory, pancreatic disease, gastric disease, liver disease, intestinal disease. And today what some scientists have concluded it can be kind of the used in case of the Alzheimer also, dementia also. So that's the latest, we can say, the innovations or latest researches which concluded. But of course, I'll tell you the one thing, that we cannot replace our anti-cancer drug with the curcumin, but at least because we have done the gamma scintigraphy studies, we have done the kinetic dynamic studies. And of course, what is my suggestion, that when we are giving the therapy of the cancer to the cancer drugs to any patients, then side by side, if you will give the capsule of the curcumin, definitely the day will come, the dose of the existing synthetic drug could be reduced. And when you're reducing the dose, side effects could be reduced. And maybe later on, that alone this curcumin capsule will work. But initially, we cannot replace your synthetic drugs with curcumin. This is what my observation is there. Of course, everybody mentioned that it can be used anti-cancer. Good enough, it is used. But we have to see at what stage we have to withdraw these synthetic drugs. So this formulation was found to be stable. Now this is the preparation, SNAC 30, which is the existing formulation which is into the market. And this is SNAC. Why name SNAC is there? That's the self-nano-emulsifying curcumin. So from the name is derived from self-nano-emulsifying curcumin, and 30 means it contains the 30 mg of the drug. And this, our formulation has got the US patent, and today the formulation is into the market by the name of SNAC 30. Even it is available in the Amazon also, and Arbro Pharmaceutical is the manufacturer of this SNAC 30. So next, please. So this is the next formulation, that is the silymarin, and we have worked with the silymarin again, solubility and the dissolution enhancement of the silymarin with fulvic acid carrier. And we have done in a simple way. We have not gone for the nano formulation. We have done existing tablets only, which is available in the market. In those tablets only, we have used the bioavailability enhancers, and with the help of the bioavailability enhancer, we could improve the bioavailability, that is 14.43 folds, and it is well combined, and our DISO profile is showing how the bioavailability has been improved. So this ICMR patent now not accepted, now it is granted. That's the composition for treatment of liver disease. ICMR has only filed this application, they were interested, and now a patent has been granted now. So next preparation is ginger oleoresin, and this ginger, you can see the capsules. So these are the hard gelatin capsules in which the formulation is filled, and as you are seeing, the green banded is there. So this green is we can say the second sealing. First sealing is okay, as we have done in the case of the solid dosage form, but the green sealing you are finding it is the kind of the sealing which will prevent the leakage of the capsules. So this is the kind of the sealing which we are giving. And when we have seen this formulation, we have carried out the gamma scintigraphy studies, and we have found this preparation is, of course, the full structure is not there of the rat. So this nano formulation is moving towards all vital organs. Again, I mean, this patent has been accepted by ICMR, and because of some reasons, I mean, again, it's not been granted yet because they have not persuaded it properly, our university. So because of this reason, I mean, it's left in between only. So this is how we have prepared this formulation. That is the ginger oleoresin. If you'll see the ginger oleoresin, it is kind of, you know, very sticky preparation, very sticky resin. As the name indicates, resin is there. So it is kind of the sticky resin which we have converted to the self nano emulsifying system, and you will be finding all the transparent preparation is there which has been filled into the hard gelatin capsule. So these were the studies which were carried out. Next is the lycopene. Next slide, please. Next slide, please. So this is the lycopene. So lycopene, we can get it from the tomato also, and we can get it from the source of lycopene is tomato and watermelon. So as we couldn't get the pure lycopene as a reference, because, you know, it is very costly reference. Initially, we have got it 5 mg, 10 mg from Sigma Aldrich, and it's costed us 40,000. So ultimately what we did is we have extracted from the tomato itself in our lab. There's the pure lycopene, and which was used for the development and the formulation of our product at the research level, and how it is been our extracts been done, tomato were taken, then pulp was there, then we have removed the skin, then seeds, and with pulp only, I mean, the extraction was done from the lycopene. And from the, you know, then which I concluded, because the dose is very small for lycopene, it is very good antioxidant, could be used for cancer, hypertension. So the main thing is, which I concluded, if we just eat 250 mg of the tomato, then we can get the 25 mg of lycopene out of it. When we have done the extraction and all weighing and all things is been done, then this factor was concluded. And again, when we have done the kinetic studies of this, so we could able to improve, and we have prepared the nanolipid carrier of this because the dose was very small. So then you have seen here 3.78 folds of the bioavailability has been improved as compared to the simple lycopene. And the simple lycopene, we have taken the sample from some pharma company which was containing near about the 7 to 8% of the lycopene. We have got it powder, so it was containing, nobody has given us like 10% of the lycopene which is normally available in the extract. So next preparation, please. This is the green tea polyphenols. There's the ECGC, and it's the main component of this green tea extract is, that is the apogalactine gallate. So this could be also used for as anti-cancer, and mainly, I mean, the this is green tea extract as the name indicates. So green tea is containing the maximum amount, and simple tea also. So is therapeutic action is in the treatment of many disorders including cancer. So we have done all the docking of this ECGC, and ultimately we have prepared 2 formulation out of this. Next, please. So now, I mean, the use of the docking I've done for many herbal moieties, but because of this reason, initially we can conclude that, because as you know, herbal moiety is used for many of the disorders, not for one. But out of it, what is the most, we can say, you are the therapeutic activity or the highest therapeutic activity of the herbal moiety is there, so that we can target it for it. So with the molecular docking or in silico studies, we can conclude this factor that our drug molecule goes to which side. Sometimes, unknown activities also come out with the moiety, that even that moiety nobody has worked, or that therapeutic activity nobody has worked, but when we are doing the docking system, we can conclude that particular other activity is also there for which we can work it. So maybe you will be getting the many research publications, for example, for anti-cancer, but maybe it is having the other therapeutic activity, like kind of rheumatoids, arthritis. So we can work on that direction also. So this is the main, I mean, the advantage of getting in silico studies. At least some kind of the things will come in our mind, that how we have to go, how we have to work. So we have worked for ECGC in two ways. One, we have conjugated with gold nanoparticles. So why we have conjugated with gold nanoparticles? Just for having the to see the anti-cancer effect of the ECGC. Why we have used the gold nanoparticles? Because the reason is it is having the good anti-cancer activity, and we have formulated into the these nanoparticles in our lab only, and that too is the nano form. When we are having not the particles in the nano form, if it is micron or higher particles, they may affect this kidney. So kidney could be affected. So that's why the good thing is we have achieved gold nanoparticles, and that will not be retained and they will get passed through it. And then we have seen the bioavailability, and the bioavailabilities when we have compared it. And of course, we have not gone so deeper for these studies. Only this much studies we have carried out, and further one can work beyond this, and can be good results could be concluded, like dynamic studies. And of course we have gone for the cell line studies, and dynamic studies could be done. And then of course the much at the molecular level we can go for these studies, and we can conclude in a better way. This is the initial, we can say, you are the data which will be giving us that the gold nanoparticles, we have improved the kinetic studies 4.47, whereas the dynamic studies have not been done yet. So next slide, please. So similarly, we have worked for the genistein. So genistein is, we know this, the soya seeds contains genistein. And of course, we have worked with the pure genistein. And then again, the genistein, the low oral bioavailability is there, 15%. Rapid metabolism, significantly reduced oral bioavailability of the drug. These were the two reasons. So what we have done, and what the disease could be targeted, breast cancer, and genistein has inhibitory action on breast cancer cell line. And this, other than this, it is, I mean, having the good arthritis activity also. Anti-arthritic, it works with the arthritis also, and especially to the women with the post-menopausal systems, these molecules works very good. So it is, I mean, maintaining the balance, hormonal balance also. So the formulation, what formulation we have done is, we have prepared the gold nanoparticles, this conjugated with drug. Then similarly we have prepared the lipid drug conjugates to improve the bioavailability of the drug. Again, not much work we have done it, and still we have started working on the liposome of this drug. And then maybe after sometimes we can conclude the results of this genistein. Still work is going on, and the formulations which were developed. Next slide, please. So these gold nanoparticles conjugated with anti-cancer drug, then the lipid drug conjugates, similar way as we have worked for ECGC, lipid drug conjugates and gold nanoparticles. And again, I am telling you, we have not carried out the pharmacodynamic studies, only the kinetic studies, which concluded that the gold nanoparticles improve 5.43 bioavailability, and whereas the lipid drug conjugates, this improve 4.53 percentage of your the bioavailability. So these were the bioavailability enhancements. So next slide, please. Similarly, worked for Boswellia serrata. For this, we have prepared the external preparation also and internal formulation also. And for the internal preparation could be used for anti-inflammatory, anti-arthritic, analgesic action. And patients with receiving this ACBA, we call as the ACBA, is that's the main constituents present in Boswellia serrata in the maximum amount. And this decrease in knee pain, increased your knee flaxation, and increased walking distance. The frequency of swelling in the knee joint was decreased. So again, much work is carried out for this. Nano formulation was prepared for ACBA, and that is we have seen. But again, much work has to be needed, carried out to be needed. So then again is you are the from cosmeceuticals point of view, again we have worked for the nano formulation, for the herbal nano formulation, in which one herbal anti-aging cream was prepared. And when we have compared with the other marketed formulation, again we have seen the much deeper effect is there. Again, I mean, we have to carry out the extensive studies for this, and which will be requiring lots of funds, like 10 to 12 lakhs of the funds if we're going to commercial lab. So that much is required, but the formulation is absolutely nano formulation, and it's water in it's oil in water emulsion, which will be having the feel of the product. And present formulations feels water when applied to the skin, and which may be converted to the water in oil emulsion. And after conversion of the water in oil emulsion, it retains moisture, retains, as we have added the humectants to it, and the giving the good moisture. And one of the industries interested to take this formulation, maybe they will launch this product. So next slide, please. So these were the some of the ingredients which were used for this anti-wrinkle preparation. That's your the Gatulin expression, then the propolis, oleoactive fly. These are all your the imported ingredients. And as I mentioned you earlier, nowadays many excipients are there, I mean, which is having kind of the extensive business. So even one can go for this also to from the herbs you can separate out the active constituents, and those active constituents could be marketed. So these are all your the phytoactive constituents which we derived from the plant material only. And we have added curcumin to it also. That's formulation only, the nano formulation of the curcumin. Then sea buckthorn oil, and this sea buckthorn oil is a good source of antioxidant. It can be used as internally also, externally also, and it's good of the, we can say, you're the photo aging agent. This is the sea buckthorn oil, and could be used in your maintaining the sunscreening formulation, having the good SPF, and we have seen that also, but the results been not concluded yet. So these were the, I mean, these form. Next slide, please. So these were the some of the formulation which I have shown on the lab scale only. So so ultimately we have concluded that our formulations are a very good results in increasing the amount of collagen, as increase in collagen directly related to reducing of visible fine lines, wrinkles. Hence we conclude that herbal formulations can be equally effective, and that too without any side effects. Then we have worked for sulforaphane. So sulforaphane is constituent of broccoli, and in small quantities it could be in the leafy vegetable, like what we call as the patta gobi. I don't know English name for this, lettuce. So maybe it is available that, but the much quantity is available in broccoli. So even we have isolated from broccoli, this the sulforaphane is just isothiocyanate, and mainly the high concentration in broccoli, and it's very good antioxidant and potent non-functional inducer, and this can be used as a anti-cancer agent. And maybe if the extensive work can be done, it can reduce the existing synthetic molecules of the cancer. It's very, I mean, that we have found it very good results of this sulforaphane. And when we have compared the oral formulation of these, and we have done kind of the cell line studies, and not very effectively pharmacodynamic studies, only the kinetic studies, and we have seen we can improve the stability, as it is not stable molecule sulforaphane. So we have improved the stability and the absorption profile of sulforaphane. Next slide, please. And again, this sulforaphane was conjugated with gold nanoparticles, and we had the plain SFNS also sulforaphane and conjugated gold nanoparticles. When we have seen the bioavailability issues, then we can see the bioavailability of the gold nanoparticles, which is just having the 4.97% being improved as compared to the plain sulforaphane. So then we have worked for, next slide please, allicin also. So again, allicin is unstable molecule which is present in garlic much in quantity, and what we have find it from the literature, that allicin activities are believed to have following positive effects: prevention of cardiovascular attacks, restore suppressed antibody responses, prevention and treatment of cancer, broad spectrum of antimicrobial activities. And as you have got it, the allicin is just mentioned, nature's gift to mankind. Not much work has been done to utilize in the clinical, owing to many problems encountered due to unstable nature, short shelf life, high therapeutic dose. But we have worked for this, and we have prepared the microbeads of this also. So our rationale was again to stabilize stable doses form, prepare the salt stable. We have worked for the ocular indication also, for the case of the glaucoma, and the much work has been carried out at AIIMS New Delhi at the ocular department. And again, extensive work is required before we declare our final results. Much of the extensive work is required for this. And these were the some, next slide please, these were the some work which has been carried out in our labs. Next slide please. So some of the herbal formulations, herbal publications to our herbal formulations, of course these are not updated your publications. We are having many more publications related with herbal formulations, and many, we have not, our research work has not been publicized much, because we want to kind of give the patent structure to it, and that's the reason Otherwise, many more publications could have been there, and still slowly and gradually. And I worked on gels also, like painkiller gels for your herbal painkiller gels, and compared with the marketed formulation. And we are just at par with the Volani and the Omani. That's the marketed formulation. And maybe the day will come we can launch this product to the market. And I'm already spoken these words to the industry people. And maybe I'm going for the development of this painkiller herbal formulation. And many formulations are there painkiller, but they never been compared with the marketed formulations, and no data is there. It is just believable that these our herbs, they are having the anti-inflammatory action, or they are good for the neuropathy pain. But unless until proper data is not there, I'm not satisfied. So I've developed kind of carried out dynamic studies out of it. Still kinetics data is required to be developed. And but I could able to judge that whatever the developed formulation is there, if we compare with the Volani dynamically, and is giving similar kind of the results, even with the curcumin also, nano formulation of curcumin. And I'm finding the good results are there. Similarly, herbal antifungal preparation, especially for nail infection, has been developed, and which I have not shown because it's the latest one that has been also developed. And again, I mean, it's giving good antifungal effect when it is carried out the antifungal activity. So my aim is that whenever you are doing kind of the research work, you should able to do in such a way that we can see the commercial aspect of this. And with commercial aspect, we should be satisfied ourselves also, whether we are satisfied whatever we are claiming, whether it is giving the good results or not. So that's my advice. Whenever we are going for the kind of the work, we should be satisfied ourselves also, that how we can satisfy just comparing it with the existing molecule, existing formulations. Maybe the bioavailability issues and other issues may be not at par with the marketed formulations, but somewhere we can say nearby this. So we can accept those formulation and we can go ahead with the formulations. So lastly, I must say that thank you for your attention and patience listening. And now whatever the questions will be there, you are always welcome. And again, I am thankful to Dr. Kulkarni and Dr. Panikumar to share this stage or share this presentation with you. Thanks a lot.
Thank you, madam. It was a very nice presentation. Sir, CVS sir.
Thank you very much, Dr. Kanchan Kohli, for sharing your thoughts on this particular multidisciplinary approach and also sharing your results. In fact, today we have two distinguished people who had supported for multidisciplinary type of activity. Kanchan Kohli combined mixed with pharmaceutics and plant products, and Harsha has done pharmaceutical chemistry as well as pharmacology with little bit of pharmacognosy, etc. In fact, this is the need of the hour for cross-fertilizing the ideas, so that some fruitful work will come out from such efforts of combining the disciplines, etc. So it's very nice. Any questions, any queries you would like to share? Chat box.
Dr. Fatima had written directly to me, so I'll share the link with Kulkarni sir.
Right.
Okay, madam.
Yeah. She said that, ma'am, can you share that link? Very informative talk, Dr. Fatima. So I'll share with you. Kanchan ma'am, it was a wonderful talk. Thank you, ma'am.
Oh, thank you. Thank you so much. Thank you so much.
Dr. Harsha madam,
Yes, sir.
is there any possibility of dividing these drugs as a prophylactic, therapeutic, etc. in terms of osteoarthritis? That is one. Other one is there any diagnostic kit? The first line of diagnosis, the second line, the third line, so that one can go from MM3, MMP3 to TNF finally and divide them into various categories.
So, sir, kit is still under development. Kit has to be developed with, everyone's effort. And hopefully, sir, the work which is done with the All India Institute of Ayurveda and Rosenberg Ayurvedic Institute in Europe, we are also combining for 1 or 2 drugs. So hopefully we'll come out of that. And definitely, sir, for osteoarthritis, and if there are tests like that right now, though, like they look out for the symptoms, and as well as they rely on MRI test more, because the other tests do not actually really and also autoimmune. So we know that like if autoimmune is expressed, it is expressed in 80% of the individuals, but especially in arthritis and all, if it is expressed and if you are having, then it's a clear indication of the onset of arthritis. So that can be used as one of the indicators in this.
Madam, is there any level in dose differences regarding antioxidant and directly osteoarthritis? Any dose levels are there? One is low and another is high, like that. Some hierarchy. Antioxidant is separate and the osteoarthritis separately, dose levels.
Yes, it definitely there's a difference, but I don't know that much of detail of that, but they are related, antioxidant and osteoarthritis. There is definitely a dose difference, because if you see as such, it is a degenerative disorder of bones and cartilage, and we are looking at antioxidant nature of herbal plants, which will be one of the factors in determining their anti-osteoarthritic activity. So but exactly what is that has to be I also need to look into that.
Can I make a bold statement?
Yes, sir.
Differentiate it. Say,
Yes,
for
sir.
example, antioxidant, the doses will be always high. But as osteoarthritis, the dose will be lesser than what is being specified for antioxidants. Looking at the target and the overall distribution, biodistribution of the drug.
Yes, sir. Yes, sir. Yes, sir. That is rightly put by you. And that's why, we need to have experts. So I have my expertise to a certain limit. Yeah. So till that limit, I'm able to answer you.
Okay, madam. Thank you. Any other questions, please? Chat box.
In the chat box, there are no questions, sir.
So then shall we go ahead with closing the ceremony?
Yes, sir, we can.
So again, as a presiding man of this particular webinar, I thank once again Dr. Harsha Karkwal and also Kanchan Kohli. Both of them have reminded me some of my friends from that particular area, going back to 20 years, 30 years. With these few remarks, I thank them profusely and hand over the mic to Dr. Professor Viresh Babu for proposing the vote of thanks.
Okay. I'll definitely tell Pushpangadan sir today. I'll call him.
Right? Yeah, yeah.
I'll call him today.
Right. Thank you very much.
Yeah. So, Dr. Fatima, I'll be sending the link to Dr. Kulkarni, sir. Then
We will share it, madam.
yeah, she's again telling, so I'm just
No problem.
yeah. I'll just send you presentation, sir. Yeah, now she
Okay.
wrote okay. Yeah.
Good afternoon, all. Good afternoon, all.
Good afternoon.
Thank you is a word which cannot be seen and touched. It can be only felt and by heart. I am very much honored and delighted to propose a vote of thanks on the occasion of this great e-conference. On behalf of my organization, I extend gratitude to our esteemed guest, Dr. Harsha Karkwal, Director, Amity Institute of Phytochemistry and Phytomedicine, for her elaborative talk on bioactive components from herbal medicine, recent advances in treatment of osteoarthritis. Madam discussed very elaboratively about various herbal constituents which can be used in the management of osteoarthritis. Madam told that they target the damaged tissue and also influence the circulatory system in improving the pathogenesis of osteoarthritis. So most of these phytoconstituents have the potential to become alternative and worthy of further development. And I would like to thank our next esteemed guest, Professor Kanchan Kohli, former professor and head, School of Pharmaceutical Science and Education, Jamia Hamdard, New Delhi, for her extensive talk on innovations related to nanoparticles of herbal drugs, where Madam discussed very elaboratively about various nano formulations of herbal drugs, their evaluation techniques, along with the applications. Madam also discussed the techniques to be employed to overcome the problems associated with the herbal drugs like solubility and stability. So I like to say that if we integrate the nano concept with the traditional medicine, then we can find a solution to conflict the chronic diseases like diabetes, hypertension, arthritis, etc. And my sincere thanks to Professor Giriraj T. Kulakarni, Principal, Gokaraju Rangaraju College of Pharmacy, for his help and constant support during the entire process. I would like to add one statement here. So during the progress, each day and every day, sir used to inquire about the progress of work. He used to discuss what problems you are facing. He used to provide us solutions. He inspired us to work constantly in order to make this event a great success and a grand success. And today it is very difficult to found words to thanks Professor C.V.S. Subramaniam Garu for gracing the occasion, for introducing the speakers, and also actively involving in question and answer session. And I like to thank all the head of the departments of our college, teaching staff, non-teaching staff, who stood by us and constantly helped us in making this event a grand success. And an event of this magnitude cannot be done in one day. The wheels start rolling months before. And we are fortunate to have been backed up by a dedicated and a motivated staff and student volunteers. So it is not possible to profusely thank each and everyone. So I thank on behalf of our organization to all the scientific committee members and the volunteers who helped us in conducting this e-conference. And finally, I thank all the delegates who present here on this e-conference. If we consider time is money, so delegates, you spent millions of money today with us. Thank you a lot. And thanks a lot for making this occasion a grand success. Thank you, one and all.
Thank you.
Thank you, Dr. Viresh.
Thank you, sir.
Thank you, Kulkarni sir. Thanks. a
Thank you,
lot.
madam.
Thank you. sir.
Thank you. Thank you.
sir.
Thank you, Kanchan madam,
Thank you so much.
for
Thank you
your
so much.
presence and
Thank you
gracing
so
the
much.
this gathering, sharing
It's
your
my privilege.
experiences. It was really nice to see you after a long time.
Yes, same here. Same
Maybe
here.
after 3 years, I am seeing you.
Oh,
Same here.
Thank you, madam.
Thank you
Thank
so
you,
much.
Archa madam.
Welcome. Welcome.
thank you, sir.
Welcome.
Thank you. Now we are closing the session, madam.
Okay, sir.
Thank you.
పని ఎగ్జిట్ చేసేస్తావా?
లేదు. Sorry. 1 second. We will open the. ఇక్కడ వస్తుందా? అబ్స్ట్రాక్ట్ బుక్ ఉందా అక్కడ? ఎవరో అక్కడ షేర్స్ కింద ఆపాలి సార్. సార్ మీరు ఆపండి ఒకసారి. You cannot start screen share while the other participant is sharing. Good afternoon to all. Hello. Good afternoon to all. Myself, Dr. Panikumar D. Anamolu. Today we are very happy and privileged to organize e-conference on recent approaches in herbal drugs and cosmeceuticals as one of the organizing committee, scientific committee member. In this regards, I thank to management of GRCP and principals are G. Giriraj T. Kulkarni and vice principals are Dr. Gangaraju and sections today sections say Dr. C.V.S. Subramanian sir and staff of GRCP. This conference purpose almost around 350 member delegates registered as delegates and around 120 abstracts received for the poster presentation section. In that, around 60 abstracts selected for our scientific committee today afternoon poster presentation section purpose. Now I on behalf of GRCP management, I am welcoming. We are warm welcome for revealing the abstract book of e-poster presentation section afternoon. And now I am opening the abstract book. This is the abstract book. Around 60 abstracts are there. Whatever choose or whatever are selected by the scientific committee members. The conference on recent approaches in herbal drugs and cosmeceuticals. So selected abstracts we formed as abstract book. This is the ad about Gokaraju Rangaraju Engineering College, GRIT. After that, our principals are message about today's conference. Then abstract put one by one. So this is the content about abstracts, whatever selected by the scientific committee. So around 60 abstracts selected by our scientific committee out of 120 abstracts. We received around 120 abstracts from other throughout India, delegates from throughout India. So finally, I thank to all delegates and abstract senders and colleges. This is the ad about our engineering college, Women's Engineering College. That is the Gokaraju Rangaraju Laila Women's College of Engineering. And this is the ad about our college, Gokaraju Rangaraju College of Pharmacy. So this is the abstracts, whatever abstracts selected by our scientific committee, 60 abstracts along with our cover page. So finally, I thank to management of GRCP and principal sir and vice principal and our section side, that is CVS Subramaniam, for giving this opportunity to me. And finally, I thank to all delegates, and finally welcome to the delegates whoever present poster presentation in the afternoon section. All the best for all participants, all the delegates whoever present the poster presentation in the afternoon section. And at the same time, I welcome to evaluators for the afternoon section, that is the poster presentation evaluation. Finally, I thank to all the delegates. Thank you. Thank you. Lunch break from 12:30 to 1 o'clock. Again, 1 o'clock, we will start the poster presentation section as per schedule mentioned by our scientific committee in the WhatsApp group. Please join all delegates. Please join by clicking on the particular link, respective link. Thank you. Thank you.
ఇవ్వచ్చు